Greetings, everyone. Welcome to today's webinar, Why Participate in Ataxia Research, Thoughts from the READISCA Study. I'm Lori Sjogren, Community Program and Services Director for the National Ataxia Foundation, and I'll be assisting with today's webinar, along with our Communications Manager, Stephanie Lucas. Any questions for the webinar speakers can be typed in the Questions tab that's found in your control panel. So today we are having a little bit of technical issues. So some of the webcams for our speakers today are not working. But we have all of our speakers here and we have good audio. So we'll proceed as scheduled with today's webinar. And also today we have Dr. Henry Paulson as our moderator for this webinar. Dr. Paulson, who is a member of NAF's Medical Research Advisory Board, is currently at the University of Michigan Ataxia Center and is a leading ataxia clinician and researcher. Dr. Paulson has been very involved with the READISCA initiative, and the University of Michigan is one of the READISCA sites. So now I'd like to invite Dr. Paulson to take over and introduce our speakers. Welcome, Dr. Paulson.
Thank you, Laura. It's very nice to be here today and nice to have you all here, over 100 people in the space here today. Thank you for coming for this webinar on why participate in ataxia research. Let me first say, as we enter into this holiday season, we hope you're all well and that you stay safe during this remarkably stressful and difficult and strange time. I know it's been hard for everyone over the last 9 months, and our thoughts are with you as you also confront this problem with the COVID-19 pandemic. But we're glad you're here today. I am delighted to have two of the experts in the area of clinical ataxia research here today to present and answer your questions. They are Dr. Tetsuo T. Ashizawa and Gülin Uz. Dr. Ashizawa is a neurologist who's worked on the genetics and the mechanisms of ataxia for many, many years. In fact, his lab discovered the first pentanucleotide repeat disorder that causes ataxia, SCA10. He's a physician who provides care for patients with ataxia, and he is the person who has spearheaded the National Consortium of Clinical Ataxia Research in the spinocerebellar ataxias for well over a decade. He was the leader of the initial NIH-funded Clinical Research Consortium for SCA, or CRC-SCA, and out of that, eventually, he led the group that is doing the NIH-funded READISCA study, a clinical trial readiness grant that focuses on two of the SCAs, SCA1 and SCA3. He also is working closely with Dr. Thomas Klockeder on the SCA Global Initiative. And our second speaker is Dr. Uz, who is a professor at the Center of Magnetic Resonance Research at the University of Minnesota and specializes in MRI and MRS imaging of neurodegenerative diseases, including the ataxias. She graduated from Bosphorus University in Istanbul, Turkey, with a BS degree in physics and chemistry, and she got her PhD in biochemistry at the University of Minnesota. She stayed there working in the Center for Magnetic Resonance Research and has become one of the international leaders in non-invasive biomarker studies, if you will, of the ataxias. She leads a program in SCAs, and they studied neurochemistry in Parkinson's, Huntington's disease, Alzheimer's disease, and ALS. She leads the imaging effort in the READISCA study, which you'll hear much more about today, that is focusing on SCA1 and SCA3, and has already published some seminal findings on neurochemical changes that occur in those conditions. So without further ado, I'll turn it over to Dr. Ashizawa, who is the head of the READISCA study, and he'll lead off, and then later Dr. Uz will follow up with some of the imaging work. So Dr. Ashizawa, I'm turning it over to you, and let me just say, I'm sorry that we cannot see Dr. Ashizawa or Dr. Uz. They are here, you'll hear their voice. I'm going to close my screen, come back on when we have time for questions. I look forward to that. Bye-bye.
Well, Dr. Paulson, thank you very much. In fact, you cannot see me, and that's blessing, probably. You don't want to see my face. It's not that beautiful or entertaining or anything like that. Now, so first disclaimer, I was born in Japan, I grew up in Japan, graduated from medical school in Japan. So I have Japanese accent. So with this heavy accent, if you have problem understanding me, just try this. Flip R's with L's and L's with R's. You will perfectly understand. Anyway, that was a very kind introduction, and I'm also grateful for the National Ataxia Foundation for this great opportunity to connect with our patients and families. Today, we will focus on observational clinical studies that do not include therapeutic interventions. In these studies, we collect data from untreated study subjects to obtain knowledge that are essential for designing sound clinical trials. Without such information, we may end up to dismiss efficacious treatments as non-efficacious ones and vice versa. There are two major observational clinical ataxia studies ongoing in the United States. One is READISCA, and another one is a CLC-SCA natural history study. And here, we'll talk about READISCA mainly. And at the end of the presentation, you can ask a question and answer, but in the meantime, you can type in and send us questions. So this slide you're seeing is a chronicle of ataxia research advances. Ataxia was first established as a clinical entity in 1863 by Nikolaus Friedreich. Guess what? Dr. Friedreich first described Friedreich's ataxia. The next 130 years, however, was a bit chaotic era of discovery of additional ataxias and intense debates on how to classify different ataxic disorders. In 1993, Dr. Zogby and Dr. Orr made a breakthrough by identifying the first dominantly inherited ataxia gene, now known as spinocerebellar ataxia 1, type 1, or SCA1. This discovery triggered a gold rush of additional discoveries of SCA genes, and that, in turn, advanced our understanding of the disease mechanism. Among these SCAs, the CAG repeat expansion mutations are the most frequently found mutation type, and we now know that SCA1, SCA2, SCA3, SCA6, SCA7, SCA17, and dentatorubral pallidoluysian atrophy, that's a tongue twister, or DRPLA, are caused by CAG repeat expansions. The CAG is a triplet genetic code that encodes amino acid glutamine. So the CAG repeat encodes a polyglutamine or polyQ repeat tract in the protein product. Expanded polyQ repeat tract makes protein toxic, so the cells get sick and disease happens. The disease mechanism of the polyQ SCAs is now understood well enough to develop rational therapeutics. Okay, next slide, please. Can you go to the next slide? Okay. Why is it so important to understand the disease mechanism? There have been many drugs studied in clinical trials, but the FDA approved none of them because of inadequate study design, a lack of convincing efficacy, excessive or unclear adverse events, inappropriate statistical analysis, etc., etc. So since SCAs are rare diseases, we must design and execute perfect or near perfect clinical trials. The ongoing observational studies will provide data necessary to design and execute such well-designed clinical trials. Next slide, please. So why is it so important? Let's see, to do this study. Now, the real title of the READISCA is Clinical Trial Readiness for SCA1 and SCA3. The goal of READISCA is to establish the readiness of international multi-site clinical trials for disease-modifying therapies of SCA1 and SCA3. We have three specific aims as shown here. Aim 1 is to develop the world's largest cohort of pre-manifest, that means before the onset, relatives of and also early stage patients with SCA1 and SCA3 by combining and expanding existing patient populations, clinical assessment data, and biofluid samples from US and Europe. Aim 2 is to validate MRI imaging and spectroscopy biomarkers. And aim 3 is to adapt innovative statistical designs and analysis of small population trials of SCAs. Next slide. So why SCA1 and SCA3? SCA1 is the fastest progressive disease among common SCAs. As you can see in the blue and the orange figure here, it progresses 1.5 to 2 times faster than SCA3. So detection of beneficial disease-modifying effect is easier in SCA1. SCA3, on the other hand, is the most common SCA worldwide, so that we can recruit more patients in order to reach the statistical power to detect less obvious efficacy. READISCA will focus on early stage and pre-manifest individuals because in the successful spinal muscular atrophy trials where antisense oligo drug, which I'm going to talk about a little bit later, in human trials showed that efficacy is much better when the intervention occurred in earlier stages of disease. Studies of animal models of SCAs led to similar conclusions. Okay, next slide, please. An important question is why participate in observational studies without therapeutic interventions? No direct benefit to the participants. So why? There is no straight answer, but I would say benefits are indirect. Namely, the study makes future clinical trials feasible and robust. High quality clinical trials makes possible by made possible by READISCA study will be essential for testing and approving new drugs by the FDA. Once approved, efficacious drugs will benefit the participant and his or her family, as well as other patients and families suffering from these SCAs. NIH is funding this READISCA study with $1.2 million per year for 5 years, namely. That's going to be $6 million altogether. Obviously, our federal government thinks it is a very important study. Next slide, please. Okay, so I'm going to talk about Huntington disease a little bit. Huntington disease is also an inherited autosomal dominant neurodegenerative disease. It's devastating as well. The disease is caused by an expanded CAG repeat that is similar to that of polyQ SCAs. The Huntington disease field is ahead of us because they had huge funding and pioneers who are interested in this particular disease. And the therapeutic development is so ahead of us. They're already conducting a large therapeutic trial of drug known as antisense oligo, or ASO for short, that silences a mutant gene product. This is a therapeutic strategy applicable to SCA and SCA3. So we are learning a lot from Huntington disease, but we are aiming to catch up with them and go passing them. Huntington disease investigators and patients have already done their homework, however. They did multiple studies of non-interventional clinical observational studies. And you can see the acronyms of those studies here. And they studied also biomarkers, fluid biomarkers, cerebrospinal fluid biomarkers, MRI studies, which Dr. Ards will talk about, PET studies, mitochondrial studies, and those are used for biomarkers. And already a large promising clinical trial and additional large clinical trials are coming. And one example is Generation HD1, and that's antisense oligo treatment trial. And so far, the preliminary data looks very promising. So we need to pursue the same kind of route. In order to do that, we need these observational studies. Next slide, please. Now, just okay. There are important questions about participation. One of them is about genetic testing. We constantly receive the question, are the test results shared with the participant? The answer is only when the participants want to know the results. So you don't have to know the results, but you have to give a DNA blood samples. But we are not going to disclose the DNA results unless you ask for it. And that is going to be de-identified. The results will be de-identified and stored with de-identified clinical data coming from your examination. And if you are 50% at risk for either SCA1 or SCS3, you are eligible. But that means 50% at risk means your first degree relative has or had a positive genetic testing for either SCA1 or SCA3. And those first degree relatives do not have to have symptoms. If they have been tested positive for SCA1 or SCA3, then you are eligible and you don't have to know the results. Next slide, please. The other common question is about COVID-19. Dr. Paulson already mentioned, and it's very scary. Yes, we are feeling a significant impact on READISCA study. Many study sites have been closed during the pandemic. Some sites have been opened or reopened, but they may be closed again because we are in the third wave. But the vaccination is coming and other treatments are being developed. So we are very optimistic. The study will be continued and we're gonna campaign to for you to participate in this study. In open centers, maximum precautionary measures have been successfully implemented and there have been no transmission of COVID-19 during the study visits. We have enrolled 190 plus subjects and the goal was 200, so we are doing pretty well. And we met NIH milestone of this study. We have about 20 subjects dropped out of the study, with one reason or the other. COVID-19 is one of them. And we'll need to replenish those dropouts by recruiting new subjects. So keep in mind, if you haven't called us, you can still call us and get into the study. Currently open study centers including Houston Methodist, University of Minnesota, University of Florida in Gainesville, University of Alabama, Birmingham, Stanford in California, UCLA, of course, in California as well, and UCSF, University of Colorado. And those names on this list may change from time to time. So this is part of my presentation. Let's continue with Dr. Ards.
Well,
Thank you for listening.
Thank you, Dr. Ashizawa. And let me just add here, I'm at the University of Michigan where we had been all ready to go. We've been approved to begin seeing research participants again. I had scheduled 3 last week, and I had 2 more this week. And of course, because of the increase in cases in this third wave, we had to shut it down. I think both our research participants and we are sad about that, but it's the right decision right now. And hopefully in the next several months, we'll be able to get things in order so that we can begin in-person evaluation again. We'll now turn it over to Dr. Oz, who will speak about imaging, which is a critical part of READISCA. Dr. Oz.
Thank you, Hank. Can everyone hear me well? Hank,
Yes.
can you hear? Okay.
I can hear you. I can hear you.
I need one response. Okay. Hi, everyone. It's great to see that close to 150 people are participating today. I'm, as both T and Hank introduced, I'm Gülin from University of Minnesota, and I have been leading the imaging piece in the READISCA study. And of course, there are a variety of imaging methods. In READISCA, we are doing only magnetic resonance imaging, and I'm going to talk to you about why you may want to participate in this portion of the study, because it is an optional portion. We are targeting to scan about half of the clinical participants in this aim. So there are various reasons, and the primary one is that MRI provides different information than the clinical evaluation. And it essentially opens a non-invasive window into the brain. And I think everyone is familiar that you can gain great anatomical information from MRI, but that's not the only piece of information that MRI provides. You can get network, chemistry, blood flow, function type information from MRI, and that is exactly what we are doing in READISCA. Then many groups around the world have shown that MRI information is actually more sensitive to the progression of ataxia in SCAs specifically than the clinical evaluations. There are various reasons for that. Clinical evaluations are obviously an essential component of any clinical trial, any natural history study, but there can be a lot of day-to-day variability. Whereas when you use an MR biomarker, an objective quantitative measure of disease progression, your variability is lower. Day-to-day variability in the data is lower. So that makes the method more sensitive to progression of disease. Also, various groups have shown, and we are investigating this question also under the READISCA, but that MRI is sensitive to changes in the brain prior to the ataxia symptoms. So in this preclinical, pre-manifest phase, the clinical evaluations obviously do not show any disease onset, but we know that changes in the brain have already started at this stage years before symptoms occur, and those changes are accessible by various MR methods. And finally, based on all this information and based on animal model data, we expect that MRI will be more sensitive to detect treatment effects in upcoming clinical trials, and that would reduce the number of participants needed to participate in these trials, which is obviously very critical, especially in rare conditions like ataxias. So I'm going to tell you a little bit what we do in imaging in READISCA, starting out with some terminology. You guys may have heard of the term Tesla, 3 Tesla, 1.5 Tesla. That designates the strength of the magnetic field that we use in the scanner shown in this picture. And very widely used clinical MRI scanners in the routine hospital setting can be 0.5 to 1.5 Tesla. But the field has been moving towards higher field MR scanners like 3 Tesla and higher magnetic field that now pretty much all academic centers, academic hospitals also have. Their advantage is to provide clearer images and more detailed information about the brain, actually about any tissue that you can examine in the body. So think about HD TVs, for example, relative to the old-fashioned TVs. You have better resolution, sharper images, higher sensitivity to detect any changes that may start happening in the brain. So first of all, the most common MR modality that's used in research and clinic is these structural images shown on the right here. Everyone's familiar with those. We do that at very high resolution in READISCA, but we also do other type of MR imaging, and that's what I'm showing here. First of all, we use the high field scanners, the 3 Teslas as shown here in this corner, to increase our sensitivity, and all our participating sites do have this higher end technology. They are major academic centers. So in addition to anatomy, we look at brain connectivity. Your brain function is supported by intricate networks, and these networks are accessible by this one MR technology that's called diffusion MRI. It uses the diffusion of the water molecules to get access to these, to the essentially wiring of the brain, and that's what we can gain information on in READISCA. Then the method that I have dedicated my career to, magnetic resonance spectroscopy, gives you information about the chemical changes in the brain. I'm showing some data. So these are the images that we get the anatomic information from, and then we select the region of interest shown in with this square here with the box, and then collect chemical information, and those data look like this. And each of these peaks here comes from a different chemical that's concentrated to give the signal, and changes in those peaks, like this one here going down relative to this individual, tells us that the nerve cells in the brain are struggling. So this is a very different type of information than just looking at the structure and anatomy. And just an overview of our imaging study in READISCA. As I mentioned, we are planning to scan half of the clinical participants, which is a total of 100 individuals. We actually reached our targeted enrollment in the study, but we need to replace, as T described, we need to replace some of those participants by new enrollees. So we still have some room for enrollment, and we focus on SCA1 and SCA3. And because we have the sensitivity to detect changes in the brain in the earliest stages, pre-manifest stages, we would like to focus on those. So a good fraction of our cohort is at that pre-manifest stage. We have 6 imaging sites in the study: University of Minnesota, Mass General, Harvard, University of Florida, Johns Hopkins, and we have 2 European sites, Bonn and Paris. And the study goes on from baseline up to 3 years with annual scans, and plus in year 1, we have a 6-month scan. Now, this is very important. So in our NIH grant for this study, we had proposed 3 scans at baseline, at 1 year, and at 2 years. So monitoring of participants for 2 years. But then Biogen came to us because they were very interested in this type of data and asked us, how can we enhance this study? I mean, this is showing industry interest because they are planning these clinical trials, which is great news. So they wanted to add 2 more time points, 1 at 6 months and 1 at 3 years. And now we are doing that together with them. And imaging, it takes about an hour in the scanner, and we collect, as I explained, data on anatomy, connectivity, and chemistry. So you may wonder, how safe is this? and what we do for imaging during the pandemic. So first of all, we don't have back-to-back scans at our centers. At our particular center in Minnesota, there's always half an hour between each scanning session so that all the surfaces that anyone, the participant, the researchers have touched, they get disinfected. I have some pictures here that includes the board, the coil, etc. And the researchers and participants wear masks, and researchers on top of that also wear the face shields. So we try to take all the precautions that we are able to, and these spaces haven't been this clean in many years, I can say. So this is just to acknowledge all our participating sites. We have 20 sites in the US, and as I mentioned, 2 sites in Europe, and displayed are pictures of all our clinical site PIs. Some of them you may be familiar with. So all these wonderful individuals came together in this study and are contributing to this endeavor. And thank you for your attention, and we are all looking forward to your questions.
Thank you, Dr. Oz. That was wonderful to get that overview, and it's nice to see the faces from investigators at various sites. I think we commented yesterday that Ohio State is also a site, and we didn't see the face there at Ohio State, but we have continued to bring in new sites over the last year and a half, and we really appreciate that. We're starting to get questions in the chat space, the question space, and it is now time for you to let us know about the questions you do have. I will start off with one that was in the chat space, and then I have some others as well. And this is regarding mentions about SCA2 clinical trials, but then the question is, will the SCA1 and 3 trials help SCA, and it has at patients? So I would say let's take that as either SCA2 or SCA patients. So that is, will SCA1 and SCA3 trials help other SCAs? And if so, what kind of timing are we talking about here? Dr. Ashizawa, perhaps you could take a stab at answering that question.
Yeah, okay. So SCA2 is a very similar disease, but different gene. CAG repeat encoding polyglutamine tract is expanded, so that part is very similar. And so antisense oligo approach, ASO approach, is applicable to SCA2. So it's a common goal in a way. We didn't choose SCA2 because we chose SCA1 and 3 as a fastest progressing SCA and the most common SCA worldwide, respectively. But SCA2 is very common in particularly Latin America, Italy, and other countries, and also United States. It's a substantial number. And earlier we studied SCA 1, 2, 3, 6, and SCA2 was not uncommon among those polyglutamine SCAs. So industry group already approached us, and they are interested in similar approach with antisense oligo or other RNA silencing technology to reduce the toxic protein. And so I can't really specifically say what company is doing what, but I can assure you they are coming, they're knocking on the door, and they are talking to NAF about doing the study. Indeed, NAF has funded similar study as to READISCA, but it's called Natural History Study of CRC-SCA. And this natural history study include SCA1, SCA2, SCA3, SCA6, SCA7, SCA8. If I don't forget anything else, I think, and SCA10. And those are included in that. And SCA2 is an integral part of the natural history study. And drug companies, pharmaceutical companies, are really desiring for the collaboration with us to get natural history data so that they can design the clinical trials. Indeed, some of the companies may have already therapeutic medications, particularly antisense oligo, and number of investigators to decipher the pathogenic mechanism have been in dialogue with those companies. So in terms of the time frame, again, I would say once SCA1 and SCA3 go into clinical trials, SCA2 will follow very closely behind, and SCA7 as well.
Thanks. Let me add to that by saying two things. One is, since SCA2 as a disease gene, has also been implicated as a risk gene in ALS, Lou Gehrig's disease, motor neuron disease, which is a much more rapid condition. There's a lot of interest in SCA2 as a target in that disease as well. So there's reasons to think that SCA2 will be as quick as SCA1 and SCA3 in getting to the table for clinical trials of disease-modifying therapies like ASOs. The second thing I want to say, and this is about the question of, does this trial help the other SCAs? And I think if you think about READISCA as being like learning how to ride a very complicated bike, it takes a while to get up on the bike and make it go, figure out the speeds, how to turn, how to use the horn, how to go from speed 1 to speed 5. We are riding a complicated bike and we're getting better as we go. And what we learn from that procedural memory is going to be useful for other SCAs, I think. And the last thing I'll say with respect to this is, SCA1, 2, and 3 are just 3 of the SCAs, when you already heard there are many more, including up to well over number 40. And there may be a need in some of the rarest forms of SCAs to take a very creative, innovative, if you will, an N of 1 approach, where people develop very specific therapies not tested in large clinical trials. But SCA1, 2, and 3 at least, and SCA6 for sure as well, are of the category where you could do a larger multi-center clinical trial. I'm going to follow up with another question, and it's kind of a two-part thing. Are there any age limitations for participation? And related to that is a question: why are we focusing on the earliest stages of disease for the most part in the study? I think I'm going to have to go to Dr. Ashizawa again for an answer on this one.
Okay, so Lori, can I share that a screen? I don't know how you do this, but
Sure.
if you have,
I'll get you to share your screen. Hang on just a sec.
I just stopped sharing mine.
Okay, great.
All right, you should be able to share your screen now.
Okay. Change present show screen, right? Okay. I don't know.
The chart
We can
looks
see
good.
your screen. We can see your screen.
You can see my screen?
Yes.
So do you see the table?
Yes, categories of enrollment. We see that.
Okay, very good. Categories and enrollment. So you can see the age is in this column of age parentheses years, and early stage patients, that category is 18 to 65. But you have to have a minimum manifestations as SARA score. That's a sort of examination score between 3 and 9.5. And that 3 is a minimum ataxia, but many normal patients, normal subjects, have a SARA score as 2.5 or less. So we said early stage is 3 to 9.5. 9.5 is sort of equivalent to ambulatory status without any assisting device. All right, so then pre-manifest carriers, the age is a little bit different, 27 to 65. So that is because we would like to see the phenoconversion, that means onset of the ataxic symptoms during the study. So a SARA score of those individuals would be 0 to 2.5, and some of them will go into 3 or greater during the study. But they are pre-manifest carriers, so that they know that they have a gene already. So to maximize the possibility that we choose this age group. Now, it's a little bit complicated, but the 50% at risk subjects. So those are your first degree relatives. They have have SCA1 or SCA3 genes. They may be symptomatic or not symptomatic, but their gene is positive. You know that. Then you are eligible to participate in the study, but age is going to be between 27 and 50. 27 is the same reason why we chose the lower limit in pre-manifest carriers, because we would like to see the phenoconversion. You become symptomatic during the study if you carry the gene. And 50 is because if you are above age 50, the chance is that you are not going to have ataxia symptoms. That is much greater. It's not completely excluded, but some people with gene carrier may have onset later than age 50. But a majority, quite majority of them will develop symptoms before age 50. So this is a 50% at risk. Half of those 60 subjects will have a gene, mutated gene, but remaining 50% will have a normal genotype, no expansion mutation. So they will not develop the disease forever. So that's the reason why those age range has been determined. And those are already closed up in the previous early stage patients. I'm not going to go into that in details. Is that
Thank
clear?
you. Yeah, that's very helpful. So again, if you don't have SCA1 or SCA3, this study is not for you or your family. If you do have SCA1 and SCA3 and your SARA score is outside of that range of 9.5, let's say your manifestations of disease are a little bit beyond 9.5, you still can participate in the study because the NAF-funded CRC-SCA natural history study is completely dovetailing nicely with the READISCA, so that you are eligible for that particular natural history study as well. Thank you very much.
Yeah.
I'm
So
going to ask.
one more
Yeah,
addition, just clarification. So that CRC SCA natural history study funded by NIF is really important, and that this READISCA study will eventually merge with CRC SCA natural history study. But at the present time, they don't do MR study, they don't do CSF study or blood collection yet, but it's coming up very soon. So
very
soon we're gonna see those.
Yeah. So
Thank
this
you. If
writing
I
the
may
complicated. Go ahead.
one more comment. Sorry, this is Gülin, for the interruption. I think also a direct answer to that question about why focus on early stage is the earlier we are able to intervene in the disease process, the better. So we want to capture those earliest changes in the brain so that we can intervene while the nerve cells are struggling, but they are not lost yet. Because once the cells are lost, you can't bring them back. But when they are sick, you can intervene and you can make them healthy. So from my perspective, that's really a big motivation.
So Dr. Oz, I'm going to take the liberty of being here to ask my own question. And maybe other people are wondering the same thing. When you talk about using your magnetic resonance spectroscopy to identify these changes in the neurochemistry of the brain, the first thought is, well, they're changes that slowly occur over time, and that if you were to give a therapy that stopped the disease from progressing, you see no further changes over time. But is it even possible that some of the neurochemistry changes could go back to more to the normal end, that is kind of revert back to normal? That's
Yes.
the first part of the question. And the second part of the question is, have we yet shown that in any good studies that when you give a disease-modifying therapy, let's say to an animal model, that you can see a change in that neurochemistry? So two parts to that question.
Yeah, yes to both. And the two are actually very connected to each other. The two questions are connected to each other. Yes, neurochemical changes are reversible. That has been shown in spinocerebellar ataxia animal models, as well as other diseases, other animal models. So we know if we intervene early, we are able to reverse some of those changes. And that reversal is detectable with the methods that we use.
Great, thank you. Here's two practical questions. If I move, will I be able to continue to participate if there is an enrolling site in my new city?
Okay, let me answer the question. Yes. So we have, as Dr. Odds showed the slides, we have 20 US sites. One of them may be very close to where you move to, and you can continue your study there. So we communicate with each other. We use exactly the same clinical examinations, testing, and so on and so forth. So everybody in the consortium are on the same page. So there is no problem, no disruption. You can continue the study.
And the same is true for the imaging portion of the study. We actually did have at least one or two participants who started out the study at one site, but then they moved and they were closer to one of the other imaging sites. So they switched the imaging site of the study. So yes.
And related to that, and perhaps you both could answer this too, is there travel reimbursement for mileage, or if you had to fly into a site to be evaluated?
Yes.
Absolutely. Yeah.
Mm-hmm.
Okay, go ahead.
Yeah,
Like
go ahead.
yes.
because we don't have imaging at all the sites. A number of our imaging participants need to travel, and that travel is covered, as well as any hotel stay that may be needed for the study at the imaging site. Yes, no out-of-pocket expenses.
So it's a little bit different story for the clinical part. Now, let's say you are living in Alaska, which is isolated within the United States, but nowhere close. Can we pay for the airfare at the present time? We don't have any budget for that. But I think we can reimburse for the driving mileage per diem, and that kind of thing is quite feasible. And even flying, if it's you have already you have been participating in READISCA and the clinical studies for a couple of years, then we don't want to lose the track. So we may fund the long distance flight to the original place or its nearby place. So that's a consideration. We can't guarantee, but that's it. That's our thought.
Thank you. As I see the names of these excellent investigators of these various universities that's up on the screen right now, I know these people are also superb clinicians, and many of our research participants are also seeing clinically at the same sites where they're evaluated from a research perspective. And the question here is, can I get clinical care at the same time that I get my research visit? That is, can they be piggybacked or in some way dovetailed together?
Absolutely. So this is a clinical natural history study, the READISCA and CRC-SCA. So either study, if you visit, already you got the examination and monitoring of the disease progression. That is going to be very important for the physicians to make judgment for the therapeutic and symptomatic treatment, particularly if you have muscle cramps or some kind of abnormal movements or something like that. Ataxia itself, we don't have any treatment, but ancillary treatment manifestations, we may be able to treat. And we can address those things as a part of the actually study if it's a minor. If it's a major thing, then we have to set up within the same visit and that different structure to justify additional testing and the treatment.
Yeah, let me say that, I think this is probably true for many of the clinical research investigators who are clinicians and also do research, is we often take our clinical hat, put it on, and we put our research hat on. And so when I'm seeing someone for this READISCA visit and I'm doing a research visit with them, what usually happens, I do the entire evaluation. Done with the evaluation, the research coordinator will continue with the research participant, and I take some time to answer their clinical questions. Sort of put on my clinical hat and do that. So even though I may not actually be billing for a clinical visit, I am, if you will, answering questions that are clinically relevant. I bet there's probably many of the investigators who are similarly inclined. Lori or Stephanie, do we have further questions? I am having trouble seeing the question box right now, so if there are others, let me know.
All right, let's take a look here and see if we do have any other questions.
While you're looking up the questions, just one thing that I wanted to clarify. And some of you audience may say, what the heck of disease modifying treatment? So that is that changing the course of the disease by attacking that disease mechanism. Now, it is different from symptomatic treatment. The symptomatic treatment is stopping the symptoms. Most prevalent and most common example is a Parkinson's disease. So you know Michael J. Fox established Michael J. Fox Foundation to find the disease-modifying drugs because he has been treated and he has been functional enough to engage in some professional and social activities, but those are dependent on L-DOPA or other dopaminergic agents, and those are symptomatic treatment. It doesn't stop the progression of the disease. So while he's maintaining, masking the symptoms, his disease progresses and eventually none of those symptomatic medications can work. So Mr. Fox is really putting a lot of effort to trying to find the disease modifying treatment. So READISCA and the CRC-SCA, of course, those studies will be beneficial for finding and improving the symptomatic treatment as well, but we are primarily gearing towards discovery of disease-modifying treatment.
Thank you. Well, I think if there are no further questions, it's been a pleasure to present this to you. We hope that you found it useful. We thank the National Ataxia Foundation for really making this possible. What NAF does for our families with ataxia and trying to understand the causes of disease and develop therapies is really extraordinarily important. If you have questions that maybe you're too shy to ask right now and you want to address them to us, we'd be happy to answer those by email or through the NAF, and we encourage you to do so. If you know someone who's interested in this study, have them reach out for investigators to see if they can participate. And we wish you well and have a wonderful holiday season.
Absolutely.
Thank you everyone for joining.
Thank you so much, Drs. Ashizawa, Dr. Oz, and Dr. Paulson for sharing all those thoughts with us on this important topic. As Dr. Paulson said, if you have any other questions, you can email us at naf@ataxia.org, or you can definitely visit the research section of NAF's website, which is up on the screen right now, for further information or for new opportunities to participate in research. Wanted to note to all of you that a recording of today's webinar will be made available for you to reference and share. And really, truly appreciate everyone joining us for today's webinar. And we hope you all stay safe. And thank you again for joining us this evening. Thank you again to our speakers.
Bye-bye.
Have a good night.
Thank you. My
Bye.
pleasure. Thank you.